It functions in some way or another to restrict cancer development. So as tumors develop, they acquire mutations in the p53 gene that prevent its activity. The interest in p53 has led to a lot of research trying to understand what the p53 protein does to limit tumorigenesis. Early studies in the 1990s suggested that p53 could be involved in DNA damage checkpoints, so that when cells acquire some type of DNA damage, either exogenously or endogenously, they activate p53 to prevent proliferation. And so one could think about what p53 was doing to suppress cancer based on the fact that it would limit cells that acquired DNA damage, and that when cells lost p53, they would proliferate and contribute to a tumor. So that was the way the picture looked in about 1992, and certainly those data are very clear in terms of function of p53. But over the last 20 years, we've realized p53 can do many other things. A number of these would be predicted to limit tumorigenesis. One is through the process of apoptosis, where p53 can respond to, again, DNA damage and even activated oncogenes to lead to cell death of premalignant cells. But another program that has been quite exciting and gained a lot of interest over the last decade is a process called cellular senescence. Normal cells proliferate only a limited number of times, after which they stop dividing and can no longer proliferate. And for many years, it was thought that this process, termed cellular senescence, was a way in which cells sort of kept the number of cell divisions they were capable of in control. We now appreciate that this form of senescence, often called replicative senescence, is triggered by the successive shortening of telomeres as cells proliferate in culture. One thing we and a number of other laboratories discovered, again, about 15 years ago, is that senescence could also be triggered in cells in response to activated oncogenes. So, for example, the RAS oncogene, when activated in premalignant cells, can trigger a program, even if the cells are young and they haven't divided extensively, that looks identical to cellular senescence, and this involves p53. As we've studied the program of senescence over a number of years, we've become to appreciate that it's actually quite interesting and maybe broadly relevant even beyond cancer. When p53 is induced, it activates downstream genes that will lead to a permanent cell cycle arrest, again, limiting cancer development. But another interesting aspect of the program is that p53, through senescence, can trigger secretion of factors that signal the immune system to target the senescent cells. So even though they don't die in a direct manner like they do during apoptosis, this indirect secretion of immune modulators can recruit the immune cells to clear out the senescent cells, again adding an extra sort of layer of tumor suppression, of ways in which cancers can be are prevented from developing. That's led to a lot of excitement about how this program of senescence is modulated, how p53 can act within the cell to prevent cancer and also affect the p53 microenvironment to modulate tumorigenesis in the hope that we ultimately can try to re-engage this senescent program in tumors. Another aspect that p53 has been involved in that has been quite exciting to me as of late relates to its ability to restrict cell fate. And what I mean by that is that it's been known now for about 6 years that loss of p53 can stimulate the de-differentiation of normal cells to stem-like cells and a particular cell type called induced pluripotent stem cells. It hasn't been known until recently whether or not this ability of p53 to restrict cellular plasticity might be important in cancer. But recent data suggests that, in fact, it might. When p53 is lost, not only are cells less able to undergo apoptosis, less able to undergo senescence, but they also become more plastic, more able to become other cell types. And we think that this may ultimately help them evolve, help cancer cells evolve various types of therapy that would otherwise eliminate them. They can sort of evade those therapies by not dying, but also by sort of becoming a more undifferentiated cell type. So altogether, 20 or 30 years after the discovery of the first p53 functions, we still don't know precisely why it is such an important tumor suppressor, although we know a lot about its biology. My own view is that, in fact, p53 probably is important because it can coordinate a whole series of anti-proliferative responses that ultimately limit the progression of the tumors. And the challenge for all of us in the field is to see, with that knowledge in hand, can we exploit it to think about new ways to treat cancer. Because p53 mutations are so common, obviously finding strategies that exploit the loss of p53 in tumors could be very broadly beneficial.